So it's my pleasure to now welcome Peter Hammerman, who's currently the Global Head of Translational Oncology at Novartis, and formerly had been running a lab at Dana-Farber, where he did very important work on FGF receptor, drug resistance, and look forward to your talk, Peter. Thank you.
Thanks, Nabil, and to the Foundation for the opportunity to speak today. This will be a treat for me, as I'll be able to talk both about the academic work that my lab did prior to my transition to Novartis, and also share some of the updated data with our experience with FGFR signaling as a therapeutic target. I want to start just by introducing FGFR's FGFR biology and the pathway. FGFR stands for fibroblast growth factor receptor. There are 4 of them in the human genome. FGFRs 1 through 3 are quite similar to one another. They're expressed across many tissues. They signal in fairly comparable ways. FGFR4 is a bit of an outlier in that it's more tissue restricted in terms of its expression and structurally distinct from FGFR 1 through 3. They're receptor tyrosine kinases. They sit at the cell membrane, and there's more than 3 decades of work detailing the biological function of FGFRs by labs really around the globe. Some of this is highlighted here in terms of the impacts on proliferation, survival, other cellular processes, but this is a core signaling pathway that's been well studied since the 1980s when the FGFRs were initially cloned. I think when we think about FGFRs in cancer, FGFRs are subject, as we heard in the talk earlier today, to somatic alterations in a number of different cancer types. They can be overexpressed, they can be amplified, so there can be additional copies of FGFRs. They're subject to point mutations, in FGFR2, FGFR3, FGFR1 as well. And we heard specifically for the case of cholangiocarcinoma about the incidence of FGFR fusions, cases where specifically the FGFR2 gene is fused to another gene in the genome, leading to aberrant activity of FGFR2 above and beyond normal. There are many different fusion partners. The biology really is continuing to evolve in terms of the function of FGFR alterations. Point estimates for prevalence are continuing to evolve. We heard more about this earlier today, but cholangiocarcinoma is about halfway down on the table there, somewhere in the range in the teens in terms of frequency of FGFR2 translocations or fusions in that disease. FGFR aberrations are also found across many other disease types. Probably bladder cancer, currently enlisted in the top, is getting the most attention just because of the prevalence, particularly of FGFR3 translocations and mutations in that particular disease. This was really already covered, so I won't go into this in much detail, but just to say that the genomics and genetics of cholangiocarcinoma is heterogeneous. There are many somatic alterations that are well characterized, some that continue to be characterized. These do not occur in isolation or in islands. They occur in patterns. We heard about some of that today. FGFR alterations are among one of many alterations which occur in cholangiocarcinoma, and just this is from a recent review written by the authors are here today, but just to state that there's heterogeneity in the genomics of cholangiocarcinoma. FGFR fusions are characterized, but one of many types of somatic alterations found in this disease. So I discussed already that FGFR2 fusions are found in a subset of patients with cholangiocarcinoma. They're found, FGFR fusions, mutations, amplifications are found in a variety of other diseases, and that prompted a number of pharmaceutical companies as well as academic investigators to investigate the biological function of FGFRs in these disease types, as well as potential modes of inhibition with a thought towards therapeutic opportunities. There are many flavors of FGFR inhibitors. I cover a few of them here. And I think my perspective on these has changed somewhat over the years. I think when I initially started working on FGFRs sort of in 2008, 2009, my impression was that we would first see brought forth multi-kinase inhibitors, which also happen to target FGFRs. So I'll show a case of a response to pazopanib later on in my talk today, but there are certainly multi-targeted kinase inhibitors out there which also happen to target FGFRs. Some of them are still being investigated, but my impression, or my personal impression, is that the emphasis on those type of molecules is waning, at least to a degree. There are a number of selective and reversible small molecule FGFR 1 to 3 inhibitors, and they tend to inhibit inhibit all three of FGFR 1, 2, and 3 because they're highly structurally related. That potentially has advantages in that some of these molecules can then be looked at or examined in tumors driven by 1, 2, or 3, but it also brings with it toxicity limitations associated with inhibiting all three FGFRs. There are selective FGFR4 inhibitors as well, developed by Novartis and other companies. I won't talk about that much today. They're principally being examined in sarcoma and hepatocellular carcinoma. And there are continuing to be innovations in developing FGFR inhibitors, so-called second generation or next generation inhibitors, some that are more potent, some that bind in different fashions, some, and we'll hear from Lippy in the next talk, potentially have activity against resistance mutations found in FGFRs which occur or come up in the context of FGFR-directed therapy. So it continues to be an open area of research. There are other types of modes of inhibiting FGFR, so not just oral small molecule inhibitors, creative approaches like trapping FGFR ligands, antibody drug conjugates, degraders or PROTACs, and as well as increasing interest in targeted delivery backbones. And really, that is to say, how might we improve the therapeutic index of these drugs by more selectively targeting them to tumor as opposed to normal tissues where they do cause some element of toxicity. Going forward. Okay, so I'll share some data and some updated data from our clinical experience to date with our homegrown FGFR 1 to 3 inhibitor. It's known as BGJ398 in some circles. We have chemical codes around all of our agents, and Infigratinib is our generic name as well. So this is a slide, and these are published data. I suspect they're quite familiar to many people in this audience, but we started investigating what was then BGJ398, now Infigratinib, in a conventional phase 1 schema. So the first and foremost goal of this study was to identify a safe and effective dose of the drug, and then to explore the drug in expansion cohorts. And this was based on the science at the time. So this was really before we knew about FGFR fusions, and we went after a few different indications where we thought that there might be some activity. So lung cancers with FGFR1 amplifications. We had a somewhat agnostic group just to try to capture patients regardless of histology with FGFR alterations. So that was the design, and we took that forward some years ago. To make a long story short, this is the activity in the phase 1 study, split here in different colors by histologies. I think the overall impression is that we saw activity in some cases. Perhaps urothelial cancer was the most impressive of the four we see here. Cholangiocarcinoma patients initially were captured in the other category, and this study was accruing as FGFR fusions were beginning to be demonstrated and documented in cholangiocarcinoma, and I think we used that information or took that information forward into further development. But I think overall, phase 1 study, some activity across different indications. My own view here, certainly not a home run in any indication here, but encouraging enough to take the molecule forward to phase 2 designs. I'll specifically talk about our phase 2 study in cholangiocarcinoma. These data were presented in a publication. Again, authors are all in the room here, or many of whom are in the room here, in JCO last year. This is likely a study that's familiar to many people in this room. It involved a pre-selection, so patients who went on the study were required to have an FGFR genomic alteration, while the vast majority of these were FGFR gene fusions. There were other FGFR alterations that also were examined in this study. The patient population were individuals who had previously received chemotherapy. We heard today about the possibilities of moving some of these targeted agents up earlier in disease treatment course, but this was a population of patients who had already received a gemcitabine related therapy or gemcitabine containing therapy, and these are fairly typical phase 2 endpoints in terms of size of the study, the endpoints we looked at. Also obviously wanted to characterize the safety and other properties of the molecule in this specific patient population. So this is again data reproduced from the paper. I won't go through this in too much detail, and my eyes aren't great, so I can't read the small print here anyway. But I think a couple of points to make here is that the patient population here is described on the left. I don't think there was anything unusual or untoward about this particular, about the particular patients we accrued to the study. The vast majority of individuals in terms of FGFR alterations harbored an FGFR fusion. This was just the flow chart for how the study was achieved: pre-selection, genotyping, and then patients who had the appropriate or had a eligible FGFR alteration were enrolled on study. And the study accrued just over 60 individuals over the course of a few years. And moving forward here, hopefully also people cannot see this in the back of the room, but whenever we do an early phase study, one of our principal concerns is the safety of the molecule. This class of FGFR inhibitors, FGFR 1 through 3 inhibitors, has a unique on-target toxicity, which is to cause elevations in phosphate levels in people who receive the drug. That happens to do with the impact or the role of FGFR1 specifically in handling phosphate levels at the level of the kidney. And I think that's important to note in that highly elevated phosphate levels can indeed become unsafe or dangerous for patients, and that required adjustments to how this drug was dosed, and also guidance to patients about dietary restrictions, use of phosphate binders, and it happened in an appreciable number of patients as well. Other toxicities on this trial were, I think, fairly, I would say, commensurate with many of our oral kinase inhibitors. So there were patients who experienced rash, patients who experienced nausea, other GI side effects. There are some particular effects on skin and nails that appear to be on-target toxicities related to this class as well. So while most patients were able while most patients were able to tolerate the drug, we did have an appreciable number of individuals shown here on the right, and even the majority of individuals who required a dose interruption or a dose reduction. So the point I'm making here is that this class of drugs has its own set of toxicities associated with it. It's not, it required in this study dose interruptions and dose adjustments for patients. So toxicity with this particular class of molecules was certainly evident, and one that requires a special attention and management. On the efficacy side, and I have an updated slide because these data are now a bit old. I think the bottom line is that in patients with FGFR fusions, at least at the time of the data lock for this particular trial, we saw an overall response rate in the range of 15%. So for patients with cholangiocarcinoma who received this drug and who were evaluable on study, about 15% of them had documented tumor shrinkage by so-called RECIST criteria, so their tumor shrank by 30% or more. There were also a number of patients who were on study for some time and achieved stable disease, and the median progression-free survival on the study, so the length of time that individuals at a median level were on the study prior to disease progression, was was in the range of 5.5 months on this particular study. There were individuals who demonstrated really much more significant benefit, were on drug for well over a year. I don't think, and I'll talk a little bit about what we're doing in the lab to understand this, and we'll hear about this in the next talk, but really I think the point here was that there were documented responses, but the responses were very heterogeneous. Some patients had long-standing responses, some patients had shorter responses, and unfortunately in this case, it was the minority of patients, even with the genomic preselection, who responded to drug. As these data have matured, and as we've continued to carry out this study and conduct this study, and this slide was generated for the GI ASCO meeting, we have noticed a favorable trend as of late, which is that the response rate has come up to about 30% now. And if you look only at FGFR2 fusion patients, so you exclude mutations and amplifications and the such, the response rate is in the high 30s. So I think this is certainly trending in a positive direction. It's not where people who in this room may be familiar with, for example, EGFR inhibition in lung cancer, or ALK inhibition in lung cancer. It's not, unfortunately, to that level, but it is trending in a positive direction, which we and others, I think, we're very happy to see. The data will continue to emerge and mature. We'll continue to update them. We have a new formulation, as many people know, that's going forward. So I think we'll continue to see where this lands, but I think this particular update was reason for some increased optimism around the potential of this class of drugs in FGFR2, particularly in patients with FGFR2 fusions in cholangiocarcinoma. And what I want to talk about for the next few minutes is really what might limit responses to FGFR kinase inhibitors. I should say that almost all of this work has been done in preclinical models, so in cellular models, in mice. You'll hear again from Lippy next about some initial evaluation of resistance in the clinic, and if anything, I talked to her last night. I think really teasing out why the responses are heterogeneous in patients is going to be really important here to understand what combination therapies might be appropriate for FGFR, for patients with FGFR fusions, what other modalities might be appropriate, and to understand even within patients with FGFR fusions who's most likely to respond to this therapy versus hopefully another active therapy. So I think there are a few just points that I want to make, and I'll illustrate them on the next few slides, principally with laboratory investigations. But I think one question is really, what FGFR alteration are you treating? Are you treating broadly an amplification, a mutation, or a fusion? Or even within those classes of alterations, there appear to be differences in sensitivity. So I think there is an opportunity to select patients better for this particular class of therapy than a sort of all-comers approach. The second, which is more of a biological question, is even if there is an FGFR alteration there, and I think this will become a poignant issue also when we consider IDH inhibition this afternoon, even if it's there, does the tumor really depend on it? So if you treat the tumor with an FGFR fusion with an FGFR inhibitor, does that tumor intimately care about that to the point where that tumor is going to shrink to a demonstrable degree, or are there other features or factors in that tumor that are going to limit responsiveness, either up front or later on? And then there's the issue that we unfortunately deal with across all of oncology, is that when we treat patients with targeted therapies, tumors do have a way of evolving over time, developing additional mutations with robust adaptation mechanisms, and requiring, I think, additional innovation on treatment approaches, additional monitoring and management, and hopefully additional approaches that are therapeutically efficacious for patients who have responded and then stopped to respond for this or other classes of therapies. So I do want to say, so I know I've been quite focused on our Novartis compound here, but I think it's important to note that we're certainly not the only company that's developed an FGFR inhibitor. We've heard about them today, and there have been some really striking cases of response. This is an individual, this is a study I was involved with as an academic with a J&J compound who had metastatic urothelial cancer. Oh, sorry, going back one. Metastatic urothelial cancer with metastases to the lung, who had, as you can see in the CT scan, spiral CT scans there, a quite demonstrable response in metastatic lesions. The second is a case report we put out some years ago from Dana-Farber. It's hard to see, but this is actually a head and neck cancer patient with a quite visible cutaneous lesion there in the neck, and that tumor really melted away in the context of 2 weeks of pazopanib treatment. The individual on the top had an FGFR3 fusion. The individual on the bottom had an FGFR2 point mutation. So certainly there is ample evidence of extraordinary responses, but I think there's really a need to understand them and refine this to a greater degree. One question that always comes up when you have questions about response rates is your drug working well? Does it hit the target, and is it effective? I think there are a couple of pieces of evidence that suggest that FGFR inhibitors are potent and hit target, though I think that an important caveat is if you have trouble maintaining a dose level in patients, you compromise to some degree potential efficacy. But this is actually work we did in achondroplasia. So to take a small digression here, people who work in germline genetics, so people who work in congenital disease, had described a couple of decades ago syndromes where patients, particularly children, were born with craniofacial abnormalities. And these children had been born with mutations in FGFR genes. And there are a number of features associated with these syndromes. One has to do with deficiencies in the ability to grow and maintain bone. So working with an academic collaborator, our translational group at Novartis looked at mouse models that recapitulate germline FGFR diseases, and were able to show that we could restore bone growth in mouse models of a disease called achondroplasia. So that was some evidence that that this drug was acting on pathway. And I think another piece of compelling evidence, which we'll hear about in the next talk today, is that patients who benefit from FGFR-directed therapies will in some cases, unfortunately, develop resistance associated with additional mutations in FGFRs that render that particular FGFR less sensitive to at least first-generation FGFR inhibitors. So again, suggesting that the tumor is finding a way around the therapy by changing or evolving in the context of therapy, but that it still has to do with FGFRs. I'll talk a little bit about this issue of what FGFR alteration or which FGFR alteration. I won't go through this in too much excruciating detail, but one of the first areas of interest was in targeting FGFR amplifications. I think that just had to do with the availability of where genomic technologies were at the time, some of the emphases of study. So there were papers from Roman Thomas's group in Cologne and Matthew Meyerson's group at Dana-Farber, which suggested that in lung squamous cell carcinoma, FGFR alterations might be quite an important therapeutic target, and reported out rates of FGFR1 amplification around 20% or so. And you certainly can find cellular models or cell lines where this bears out. So one of the cell lines many people study is this NCI 1581 on the top right, so showing very significant potency of FGFR inhibitors there. But when you looked across cell lines that had FGFR alterations, and I'll get to this in more depth, it was a bit of a hodgepodge. So you found a lot of cellular models and later animal models with FGFR alterations, but there wasn't necessarily in any way a clear correlation among FGFR alterations and response to therapy. And I think these numbers, even from panel A here, which had 3 out of 4, I think it's more 2 out of 2 now. So at best, it was maybe about 50% predictive. We did take BGJ398 into lung squamous cell carcinoma. This PET scan on the bottom left was one of our better responders, so there wasn't a tremendous amount of activity in this particular population. And even though you could see, and I think what we saw is that there was really a breakdown of the relationship among FGFR amplification, pathway, activation, and dependency. So I think the point I want to make here is that while a tumor may harbor an FGFR alteration, that doesn't mean with any, very, very high level of certainty that that tumor will definitely depend on FGFR and definitely respond to an FGFR inhibitor. Taking this to the next slide, I think this is also the case with FGFR point mutations. This is work from my old academic lab where we saw this observation in cell lines and in the clinic that there was certainly not a robust correlation in some cases among FGFR alterations and response to therapy. And we said, okay, was this just something we were getting faked out by in cell culture? Was this potentially an artifact? Can we actually define and describe which FGFR alterations really are cancer causing versus ones that might be modifying or not as strong? So we marched through using FGFR2 as our model there, making mouse models, and we did FGFR3 later, of basically almost all of the FGFR variants we could find. And this was a collaboration among my lab and Kwok Wong's lab. And what we generally found was that a subset of FGFR alterations clearly were sufficient to cause tumors and cancers in a mouse model. These tumors responded very nicely to any number of FGFR inhibitors, but the tumors took a long time to form. They didn't form in any mouse, and in some mutations, they form better than others. Again, suggesting there may indeed be a need for a second hit or other alterations that are important in the context of FGFR-driven cancer. And what I mean to say there, and I think, potentially discouraging for monotherapy approaches, I think it begs the question of what are combination approaches that are going to make sense here when you might have an impact of FGFR inhibition, but by no means a complete shutdown of tumor activity. And those were just some studies we did preclinically. we've tried to take that a little bit further, and this is just another way of demonstrating the same concept. What we did here was to look across all of the tumor models we had. This is principally cell lines, but we've done this with patient-derived xenografts and the such as well, and to basically profile all of them for sensitivity to, in this case it was BGJ398, but it could be done with other compounds as well, and say how well can we develop a correlation among FGFR sensitivity and FGFR dependence. And there is a correlation. It reaches statistical value. If you have an FGFR aberration, you're more likely to depend on FGFR, to be sensitive to FGFR inhibitors, but it's not a perfect correlation. And what I think is really important for us to understand are, if you look at the second row here, the tumors in the middle or the right here, which have FGFR alterations, but at least by giving singlet FGFR therapy are not responsive a priori. We've done a number of studies, I'll show some of them in the next couple of slides, looking at what combination opportunities we might want to pursue to really try to get response rates up and improve outcomes for patients who have FGFR alterations but are not achieving dramatic responses to therapy. So this is a hodgepodge of a few different publications. Some of which are, one of which is published, one from my academic lab, which is coming out, and then one we have, I believe it's actually on the next slide, from Novartis. But we looked at this in a few different ways, and I think this is just one approach that can be taken. We looked at preclinical models, so cellular models and animal models, and also at patient-derived specimens to ask, what might be blocking or what might be an impediment to responsiveness to FGFR inhibitors, and did this with a few different approaches. The first approach that we took was simply to look at what in terms of the playing field might rescue a tumor that's sensitive to FGFR inhibitors to FGFR inhibition. So basically, are there molecules that are present in the circulation that we think could render a tumor less responsive to FGFR inhibitors? And one of our big hits from there is a gene called hepatocyte growth factor, or HGF, which binds to the MET receptor, which has been implicated in resistance to a number of other compounds. And through additional analyses we did looking at cellular models of FGFR-driven disease, we were able to report that that even in quite responsive models, MET can drive resistance in some contexts. So that's a combination that we've looked at in some depth. We took a genetic approach here in addition to a secreted protein response approach, and just asked across the whole genome what genes, when they're expressed in a tumor, can render a typically FGFR sensitive tumor resistant to FGFR inhibition. And we saw some of the usual suspects. We heard about the RAS proteins this morning. Those were almost sort of our positive controls for the assay. But two pathways that came out that were really of interest to us were the so-called TAM kinases, tyrothrioxyl and MERTK, and the NTRK kinases. And those are interesting for a couple of reasons. One is that we have good drugs or evolving drugs that target these pathways. So there would be potentially therapeutically tractable. And second is that in an unbiased approach, looking at simply what's happening in tumors that are exposed to FGFR inhibitors at a transcriptional level, we saw that these pathways are upregulated in the context of therapy. So basically providing additional rationale for some combination approaches here, really with the goal of pushing up response rates as much as possible. But I think this led to two independent conclusions. One, and we'll hear about this more in the next talk, is that tumors which respond can become insensitive due to acquisition of additional mutations in FGFRs. There's one approach you would take there, but that also that parallel pathways and compensatory pathways are relevant and really sort of beg the question about combination approaches. We've modeled some of these in cell lines and in animals. This is just sort of a colony formation assay here done on the left, where we've looked at combinations that have been nominated from our studies of clinical material, our studies of cell lines in animals, and I think we have a set of prioritized combination targets that we would be interested in exploring further. One of which I think that's been particularly interesting to us, and based on not only our work, but on work done by others, are combinations of FGFR and EGFR inhibitors, provided we can manage the toxicity. And that we were able to arise at both from sort of an unbiased approach that I described in the past, and has also been interesting in that tumors that are driven by EGFR, principally lung cancers, may use FGFR as an escape mechanism. So this crosstalk may extend above and beyond FGFR-driven tumors, and something that we're looking at in more depth now. So with that, maybe since I'm right on time here, I'll summarize with a few statements here. I think somatic alterations in FGFR are found across many tumor types, probably about 15% of cholangiocarcinoma, though that number continues to be refined. Many companies are developing FGFR inhibitors. That's likely well known to the audience here. Our FGFR inhibitor demonstrates activity in FGFR-altered cholangiocarcinoma, and our preclinical data suggests that there are opportunities to really build upon efficacy of FGFR monotherapy with hopefully increasingly effective combination approaches. So with that, I'll thank everybody here and end on time. Sure.